I will introduce today what we call the red blood cell clinic that represents a platform of innovative tools for the analysis of red blood cells. So, red blood cells are our favorite cell, of course highly elastic and are highly deformable and flow through vessels narrower than old size. And when red blood cells deformability, I have to cut the Wi-Fi because the computer search for the Wi-Fi. So. I don't know. Sorry, I don't know. How can I do? No. So red blood cells are deformable, and when red blood cells deformability is altered, there is a high risk of red blood cells blockade in the spleen, as Anna Christina said, and also in small venules and capillary. A number of pathologies with hemolytic anemia are associated with altered stability, deformability, viscosity, as well as mechanical and functional properties of the red cells. So, we developed recently in Paris, thanks to our INSAM unit, the National Institute of Blood Transfusion, the JEREX, and the Imaging Institute, an integrated platform allowing membrane expression of surface proteins, morphological, mechanical properties, as well as functional adhesion and transport properties of the red blood cells. And to do this, we used conventional technologies as well as innovative technologies marked in brown, such as imaging flow cytometry, phasic system, or microfluidic tools. So, in the near future, our dream in the JEREX and our challenge is to be able, from a blood sample of patients exhibiting red cell pathology, to perform all this type of analysis in a very fast time. And so to do this, we develop, in addition to conventional flow cytometry or SDS electrophoresis technologies, other new technologies as LORCA, imaging flow cytometry, microfluidic devices, phasic system, dynamic adhesion system, and so on. So I will go in more detail further on. So the LORCA apparatus, as the mechatronics company said, is the all-embracing solution for hematology research in red cells, able to perform measurements of red blood cells deformabilities using laser diffraction ectacytometry, measurements of red blood cell stability, as well as red blood cells aggregation and disaggregations, but using fresh blood samples. It's not possible using freeze blood samples. So we also recently developed, in collaboration with the l'École nationale, l'ENS Cachan, microfluidic approaches to investigate as well red blood cell deformability and elasticity in human pathologies. So in order to identify first rigid red blood cells and evaluate the abundance in a cell suspension, and also to measure the effect of different mechanical constraints on red blood cells and elasticity and resistance to hemolysis. So we first designed with our colleagues in the ENS Cachan spleen-like motif with a biochip consisting of polymer of microchannels obtained by soft lithography. And as you can see here, this microfluidic device mimicking the diameter of the spleen is composed of microchannels with obstacles here separated with wears of 5, 4, 3, and 2 microns. Merci. Yes, okay. So to validate the system, we first mixed heated and non-heated red blood cells. Oh, sorry. So it's the Wi-Fi. Heated and non-heated red blood cells. So heated red blood cells labeled in green and non-heated control red blood cells in red. And we pass these blood samples inside the flow, cooked with the microchannels. And as you can see here at different times, only the rigid red blood cells labeled in green here. And here, I don't know if you can see with the light, but only the rigid heated red blood cells labeled in green are stopped, are blocked at 2 microns, mimicking the diameters of the spleen. So secondly, we performed exactly the same experiments, but with Plasmodium falciparum infected red blood cells, and with two populations of red blood cells, in trophozoite stage, labeled in green, and ring stage, late stage, labeled in blue. As you can see, same results were obtained with late Plasmodium falciparum infected red blood cells, and only late stage Plasmodium falciparum blocked at 2 microns. So interestingly, we recently performed exactly the same type of experiments with some red blood cells from HS patients. So HS patients are labeled in green, and control patients in red. And you can't see with the light? So it's better. Yeah. Yes, you can see the green and the right red blood cells. So patients in green and controls in red. And in parallel, in Paris, membrane diagnosis are performed in Robert Debré Hospital by LORCA, by ectacytometry. And very interestingly, you see that there are some retention of red blood cells in green from HS patients, from those, only those having the most altered mechanical properties measured by ectacytometry. So I'm sure that Anna Cristina and Priscilla and Dimas will be very interested by this alternative technology, because we can visualize directly the blockade of the cells. So next, we also recently designed with our colleague another biochips mimicking microcapillary and venules of 5 microns. And we performed some experiments with, of course, sickle red blood cells. And so we labeled sickle red blood cells in green and control red blood cells in red. And as you see at different times during the flow, in time 2 and 3, there you observed a vast majority of sickle red blood cells in green that blocked at 5 microns, so mimicking the diameters of small capillaries and venules. So finally, we very recently designed a so-called stretching motif of 5 microns, where one channel here is composed of 6 repeats of 5 micrometers, and the chips is composed of 25 repeated motifs. And this stretching motif permits the analysis of deformability, elasticity, and endurance of red blood cells. And also the time for the red blood cells to recover its shape could be quantified. And yes, if it works, I have some preliminary results movie. So here you can see in these biochips the flow of normal red blood cells. Sorry. And you see that red blood cells are highly deformable and could flow into 5 micron and they recover the shape. When performing the same type of experiments with sickle red blood cells, you can see the difference in the shape. So you have a blockade with very rigid sickle red blood cells, and you see some sickle-shaped red blood cells. They could go through 5 microns. So very sickle red blood cell could pass. So I think that it's an interesting method to visualize, to analyze the mechanical constraints of sickle red blood cells into the capillaries. So image streams, AMNIS, is a very powerful technique that combines a camera and a conventional cytometer, allowing to obtain images of different events observed in conventional cytometry. And you could perform a lot of analysis as co-localization, cell-cell interaction, and even analysis of morphology of the cells. And indeed, flow cytometry allows quantitation of probes on many cells, but zero spatial resolution, and of course no location information. In contrast, of course, fluorescence microscopy allows good spatial resolution, but measurement of only a very small number of cells. So this technology combines the performance of both the technologies. And for example, you could analyze cells morphology, as you can see, by calculating the circularity scores. And there is an example for the company that you can see the difference in the morphologies here in monocytes. And a few years ago, using these technologies, we performed with a blood sample of sickle patients gradient density, and we isolate the different cells populations. And you can see that by labeling red blood cells by anti-glycophrine A and white blood cells by anti-CD45, we are able to visualize aggregates between red blood cells, 3 or 4 red blood cells and white blood cells. Here are 2 red blood cells with 1 white blood cell, here 1 and here white blood cells with red blood cells microparticles. So with this technology, we were able to visualize the presence of red blood cell leukocytes aggregates in sickle cell patients. So very recently, a so-called quantitative phase imaging, the phasic system of the SID4 biosystems, is a technology based on a patented technology of measure of phase. And this technology improves strongly, as you can see here, the contrast of the images of biological samples, allowing their observation without any staining, and allows quantitative image analysis. And these systems is useful for cells counting or tracking, and also to measure, and I think that it's the only system that could measure a dry mass of cells populations. And you can see just an example, sickle red blood cells with conventional images, and with the phasic system, you can see that it could reveal red blood cells with heterogeneous content, and this without prior labeling. And also, it could quantify the dry mass and the distribution. And it could be very useful to maybe to label certain clinical manifestation in the disease. Sorry. So, as Ada Cristina says, we developed a few years ago a dynamic adhesion platform, and this, as you can see, this platform in the National Institute of Blood Transfusion is equipped with an inverted microscope coupled with modules of gas and controlled temperature, so you can perform adhesion experiments at 37 degrees and at a different level of hypoxia. And the fluidics consist of nanopumps from Celix connected to multi-flow, and we can perform today 8 experiments in parallel using the Celix biochip, in which the 8 channels are either coated with purified protein or here with a monolayer of endothelial cells. And just to summary, all our results performed with this powerful platform. We demonstrated that abnormal adhesion of red blood cells to leukocytes and to endothelium could play very important role in vaso-occlusions or thrombotic events, of course in sickle cell disease, but also in HS patients, as well as in malaria, in diabetes, and also in non-hematological diseases as Gaucher disease, central retinal vein occlusion, or polycythemia vera. And of course, we search for red blood cell adhesion blocking molecules that could be very useful for additive treatment of sickle cell disease, but also in various hematological and non-hematological diseases. So we have also some apparatus permitting functional characterization in as a stop flow that allow to measure very rapid transport kinetics, for example, gas or ions. And also in European Georges Pompidou hospitals, very near to our unit, an intravital microscopy allowing to visualize in vivo in murine models, for example, adhesion of or blockade of blood cells. So thank you very much. This is the human resources, and thank you very much to our unit, to the National Institute of Blood Transfusion, and to the GREX and the Imaging Institute.